Greetings, everybody, and welcome to the last day of NACFC 2021. I'm Beth Kramer from Cincinnati Children's Hospital, and on behalf of myself and my co-chair, Dr. Andres Raab from Emory University, who will be working behind the scenes, I'd like to welcome you to workshop session 31, Difficult to Treat CFTR Mutations and Novel Modulator and RNA Editing Technologies to Fix CFTR. We've heard a lot this conference about how highly effective modulator therapy is changing the disease course for many people with CF. However, for around 10% of people with CF, such highly effective therapy is not available. For these rare or difficult to treat mutations, we must discover innovative techniques or repurpose existing therapies. This session will consist of 5 15-minute talks. Each talk will be followed by an 8-minute live question and answer session with that speaker. Please type in your questions to the Q&A box to the right of the screen and feel free to upvote any questions that you would also want to hear answered. So we will start relatively narrow in scope, beginning with an in-depth discussion of a single mutation and move on to more broad topics as we progress. So to start us off, we have Dr. John Brewington from Cincinnati Children's Hospital discussing pharmacologic rescue of the N1303K mutation.
My name is John Brewington, and I'm a pediatric pulmonologist here at Cincinnati Children's Hospital. I want to say a special thank you to the program committee and also to the moderators for this session for inviting me to talk today. I'll spend the next few minutes thinking about pharmacologic rescue of a specific CFTR variant, N1303K, across a number of different laboratory model systems. My disclosures are shown here. I will specifically point out that I am going to talk about off-label use of CFTR modulator drugs during this talk. I want to start in the same place that I'm going to end today, and that's with a huge thank you. The data that I'm going to share was generated across a number of different labs. The people and places are shown here, and it was a huge effort. And so I'm very excited to be able to share this. I appreciate everyone by allowing me the opportunity. Now, for background, I want to start in a familiar place for most of us, which is that highly effective modulator therapy is available for the majority of our patients with cystic fibrosis in North America. But if we consider those who do not currently have an FDA-approved modulator, we know that there's a cohort within this group that would have benefit from the existing drugs, but simply has no path to drug access. Many, including our group, has proposed that you can overcome this access gap through different laboratory models. Heterologous models, such as Fisher rat thyroid cells, have had great success in these efforts. This is the way in which we've had label expansion of FDA-approved modulators across a number of different drugs. These models have high reproducibility. There are also a number of patient-specific models, which have very clear focus on the individual patient and perhaps higher precision to that subject than the heterologous models. Both of these are strong approaches. Now, again, today I'm going to focus on one particular CFTR variant, N1303K. This has been reported, especially in people who have Mediterranean descent. And in the North American CF cohort, we see this in about 2.5% of our patients, making it the 5th most common variant, at least as reported by the CF patient registry. This produces a missense variant that's in the 2nd nucleotide binding domain. And the protein defect is one of trafficking, very similar to what we see with F508del CFTR. And this is shown in the Western blot on the top right here, where compared to wild type, N1303K protein does not have a mature glycosylated C-band, implying that it doesn't reach the cell surface. Now, since the defect is similar to what we see in F508del CFTR, it's logical to wonder if the same corrector compounds that improve F508del might have activity for N1303K. Unfortunately, a number of previous studies, including the one shown on the bottom right with electrophysiology data, have not held this to be true. We've not seen benefit from either lumacaftor or tezacaftor in this particular variant. Now, my personal entry into this question started with some nasal cell therapy work, which we do a lot of in Cincinnati. We enrolled a single subject who had mild lung disease, was pancreatic insufficient, and who carried a single copy of N1303K, as well as a G27B CFTR variant, which is effectively uncharacterized. And we asked, could this subject benefit from our available modulators? Now, these data were reported last year at this meeting, and so I'll cover them just very briefly. We were able to grow these nasal cells that were obtained locally into our two primary models, air-liquid interface and spheroid or organoid cultures. And as shown on the right, when we quantified CFTR function in these models, we saw a really robust correction and improvement in CFTR function when the cells were treated with elexacaftor, tezacaftor, and ivacaftor, which I'll refer to as ETI moving forward. And in fact, the response that we saw in these cells was fairly similar to what we see in F508del homozygous cells when treated with ETI. Now, these data were used to obtain off-label approval from the insurance for this subject. And he was able to start on the drug. Unfortunately, this led to several start and stop periods because of both liver and gallbladder disease, including at least one hospitalization and at least one abdominal surgery. And so in that context, as you can imagine, the clinical response is a bit mixed. But we can say that on this drug, the patient had stable lung function despite these admissions and surgeries. His BMI remained stable. His sweat chloride compared to his diagnostic test dropped significantly. And he had a cessation of a number of respiratory symptoms, including daily sputum, frequent hemoptysis, and no formal pulmonary exacerbations. And this led us to believe that we may see benefit for this drug in people harboring N1303K CFTR. Now, at the same time, a number of other groups were studying this, and several published reports have come out since that look at this particular question. And this covers a number of different models, including cell lines such as HEC293s, nasal cells, as we had looked at, and bronchial cells. The number of donors in this pool are relatively small, but the data are consistent. I'm showing some data here from the group in Toronto, showing both protein level correction in the Western blot on top, and also electrophysiologic correction, and two chamber tracings on bottom, of N1303K homozygous nasal cells when they are treated with ETI therapy. At the same time, our group became interested in gathering an increasing number of subjects to ask, is this consistent across different people? And so we were able to gather a group of N1303K heterozygous patients in our study. And also at the same time, investigators from the RARE trial have had a vested interest in this work. And so we were able to get together with them and generate the data that I'll show in the next few slides. These data cover numerous different model systems, and I'll walk through them, starting with those that are perhaps the furthest removed from the human patient, and move to those that are perhaps the closest to the patient. The first of these models should be familiar to most people. These are fish or rat thyroid cells, which have been very effectively used in our development and our expansion of CFTR modulator therapies. These data were generated by the folks at Emory. If you're interested in more information about their particular approach to FRT data, be sure to check out poster 619 and stay tuned in this workshop for a presentation on how they approach this model. Now, for this particular question, a stable N1303K CFTR expressing FRT cell line was used. And as you can see in the individual tracings and the aggregate data shown on the right, we saw a really impressive response to ETI therapy in these cells. And this is about half of the response seen in F508del expressing cells, which brings the total CFTR function up to about 30% of wild type. It's a very impressive improvement. Now, just to link back to the clinical information that I shared with you, remember the other variant that our patient carried was a G27V. And I will tell you that work with this group that again was presented last year shows that that variant is also quite responsive to elexacaftor, tezacaftor, and ivacaftor. So that clinical picture is a little bit muddy. In addition to the electrophysiologic data I just shared, we also have Western blot data here showing an increase in total CFTR production, perhaps an increase in the mature fraction when cells are treated with elexacaftor, tezacaftor, and ivacaftor. The next model I'll share with you is data that's coming from Bethesda with the Cystic Fibrosis Foundation Therapeutics Lab. This model system is going to be the 16HBE-GE cell line. These are gene-edited parental 16HBE14O- cells. For people who are familiar with that cell line, this is a bronchial epithelial cell line that's been immortalized. And the gene-edited portion is particularly fascinating here. In this model, they've created 2 copies of N1303K CFTR in the genomic CFTR locus. And so the expression control is what we would expect to be the native expression control in human cells, as opposed to, for example, artificial promoters often used for other model systems. And this is particularly exciting and has been well described in previous reports. To the right, I'm showing an example of wild type and F508del homozygous cells created with this model. The analysis here is looking at chloride conductance through a machine called transepithelial chloride conductance measurement, or TAC. When the group at the CFFT lab created these N1303K homozygous 16 HBEG cells and tested them against elexacaftor, tezacaftor, and ivacaftor, we again saw an impressive response. Just to orient you, the two rows of data on the top are cells treated solely with elexacaftor in increasing concentrations from left to right. And on the bottom, cells that have been treated with those same increasing concentrations of elexacaftor on top of a background of a stable concentration of tezacaftor, or VX661. And what I hope you can appreciate as you follow each row from left to right is that the CFTR function increases significantly. And comparing top to bottom, the function is additive when VX661 or tezacaftor is included in the mix. Graphically in aggregate, these data look like this. Black bars being VX445 or elexacaftor by itself, and gray being the combination of tezacaftor and elexacaftor. With this, the maximal stimulated amount of CFTR function reaches about 20% of wild type in this model. And there is, again, additive function between tezacaftor and elexacaftor. So continuing data to support the idea that N1303K CFTR is responsive to elexacaftor, tezacaftor, ivacaftor. We'll continue to move closer to the native patient tissue. In this case, data from University of Texas Health Sciences Center in Houston, whose group has done incredible work with taking induced pluripotent stem cells, in this case generated from skin fibroblasts, and differentiating those forward into airway epithelial cell cultures. That process has been published. Here's an image from that paper. If you're interested in this model, be sure to swing over to poster 667 and also check out workshop 14, where this model will be discussed in more detail. In this case, the iPSC-generated differentiated cells came from a subject who had one N1303K and one W1282x. And we were able to analyze these inducing tumors. Individual tracings are shown here on the left. What I'll highlight is the control tracing in black and the elexacaftor, tezacaftor, ivacaftor treated tracing in red. And there's a significant difference there. Taken in aggregate, the data are shown on the right with a drastic improvement in CFTR function. Now, we don't have normative data from wild-type subjects to understand what fraction of normal this is, but the absolute increase in CFTR-specific current is rather large. We'll now move to primary cells, in this case, nasal cells. And first, I'll show you data from a single subject who's homozygous for N1303K. Now, these cells were a kind gift from the folks in Toronto to the team at Emory, who grew them at air-liquid interface and analyzed them in Usum chamber. And what I hope you can appreciate from the raw tracings and the quantified data here is there's a significant improvement in CFTR function when cells are treated with elexacaftor, tezacaftor, and ivacaftor. Now, this is a small number of samples from a single subject. We don't have normative data to fractionate this as a percent of wild type, but it's still promising. Our group in Cincinnati had gathered also a group of N1303K heterozygous patients. These cells were all obtained at the local center and shipped to us. And there's a number of different mutations that are held in trans for these subjects. In the graph on the right, you can see these mutations listed at the bottom. I'll highlight that the first 5 are all generally considered missense mutations. So the second variant may be in play in any response that we see. Whereas the subsequent 6 are generally considered not to make significant protein, either because of a frame shift, a canonical splice variant, or a preterm termination codon. Across the board, though, 10 of these 11 subjects had some form of improvement on therapy with ETI in the lab. And of those cells, 9 of the 10 had statistically significant improvement. Only 1 showed a decline. Now, the average improvement was fairly significant, about 10% of wild-type function, but this is skewed heavily by 2 pretty robust responders. Our first subject was a 5T, 12T gene trans, and then the G27B subject that I've already mentioned to you. Regardless, though, there is consistent improvement across all of these heterozygotes, with one exception, when the cells are treated with ETI, continuing to support the fact that this is a biologically active compound for the CFTR variant. So I'll end here. Again, hopefully I've taken you through a number of emerging heterologous, modified human, primary human models that we can use to study CFTR variants. And when we think about N1303K, we've shown across a number of these models, both in published data and also in what I've shared today, that there's consistent benefit from elexacaftor, tezacaftor, and ivacaftor. And this is perhaps more convincing because of the wide range of methodologies and tissue sources. This response is a little less when comparative data is available than what we see in F508del, but it's still significant. And we believe that the next logical step here is a clinical trial to understand the efficacy of this drug for this variant. This clinical trial ideally would include both heterozygotes and homozygotes because we don't have good comparative data to understand if that response is different between the two groups. I'll end here again with a huge thanks to everybody who's been involved in this work. I appreciate your time. I look forward to answering any questions that you may have.
For the tour de force, John, on the excellent work going on as we look into these mutations. We are aware, sorry folks, that there was some choppiness of the video, and hopefully that will resolve. So to kick us off, John, I have a question for you. So cell monolayers derived from healthy individuals can display a wide variety of CFTR activities. How is the activity level of wild-type CFTR established to compare N1303 correction? And what are your thoughts on how many different patients are necessary to establish such a threshold?
Hey, thanks, Dr. Kramer. And I'll also echo my apologies for the video issues, but it's a very good question. And in the case of the data that I've shared today, the establishment's been a little bit different across each lab. So I'll speak specifically to how we've done it with nasal cells in Cincinnati, so I don't misspeak on anybody else's behalf. In our case, certainly in the primary cells, there is definitely wide variance amongst wild-type CFTR function when you quantify it. And so I don't know the exact right number for what the N needed for normative data is. We've settled in our group on a cohort of about 15 different wild-type subjects and have focused on the confidence interval and getting that as narrow as possible. But I don't know what the right answer. I think the real question that I, again, also don't fully know the answer to is, even if you know what wild type is, how much wild type function do you need to make a difference? And again, a number of about 10% has been brought up from a number of different model systems. That's the cutoff that's been used in the label expansions based on FRT models. I don't know that necessarily translates from model system to model system. But I think that's the real crux of the question, is once you have that normative data, what do you do with it, and how do you apply it to the data that you generate for a patient with a rare variant?
I think that's a very, very interesting point and a complex question. And I guess, not to put you on the spot, but what are your thoughts about ETI approval for folks with at least one copy of N1303K? And I know you called for larger clinical trials. What are your thoughts on approval for it if you had the power?
Yeah, I mean, I think at this point, Dr. Kramer, we've got consistent data across a large number of model systems. It's clear that there's definitely some degree of improvement. The real question is, again, is it enough improvement? I suspect, looking at what we have, that the answer is going to be yes. That said, the traditional approaches for label expansion, the FRT libraries that Vertex and colleagues have put forward, did not sort of meet whatever metrics or criteria were expected for that. And so the traditional label expansion approach is not happened. And so the next logical step for us to see if there's benefit would be a clinical trial. But again, I think that the data that's available to us that we have is consistent across a large number of model systems, a decent number of patients, that there is some degree of benefit here.
Yeah, I think that consistency really came across and really speaks to the power of these systems, especially as we try to reach the remaining folks out there who don't yet have highly effective therapy approved. Fantastic. Well, thank you, Dr. Brewington. We will move on to our next speaker, Candela Manfredi from Emory University, who will be speaking on factors that influence CFTR modulator response in cell culture systems, a very pertinent topic. We are moving on to the second talk of the session. Again, this is Dr. Candela Manfredi from Emory University, discussing factors that influence CFTR modulator response in cell culture systems. Thank you for your patience.
Hello, everyone. Thank you for the opportunity to present my work. The title of the talk is Factors that Influence CFTR Modulator Response in Cell Culture Systems, Predictive of Clinical Benefit. There are no relationships to disclose related to this presentation. So in this first slide, we take data from many laboratories, indicating that the parameters sweat chloride and short circuit current data measured in vitro, tracked together with the clinical phenotype, lead to the hypothesis that all these parameters can be used to predict clinical response to modulator treatment. So in this talk, we evaluated and we want to address some of these assumptions. In this slide, we observed the relative CFTR mRNA expression from primary airway cells separated by type of mutations and wild type. And on the very right side of the plot, we observed nonsense mutations, in this case, W1232x homozygous. And we see that the CFTR mRNA gene is expressed in a very low level. And this is due to the action of NMD pathway, which is degrading mRNA. But on the very left side of the plot, we observed the expression of the wild-type CFTR. And we can see that the gene expression could be quite variable, and the variability can be by several folds. And this plot, again, we show in the mRNA expression of CFTR in, again, wild type and patients with CF in airway cells, and all together next to the measures of the short circuit current. Okay, and we observe again that big variability in mRNA expression of CFTR among all these patients, and that the magnitude of CFTR activity can be quite variable too. And this is important because it has been used, the 30-40% of wild-type CFTR activity in primary cells as a threshold to predict improvement in patients with CF. But what we see here is that this magnitude is very variable and is very significant, the variability among the wild-type individuals. So we correlated the two parameters, the measure of CFTR mRNA and the CFTR activity, and we observed they correlate. And with this observation, we conclude that we should consider these two parameters, the level of mRNA and the measured short circuit current, because they can impact in the likelihood of patients to respond to certain type modulators. Another factor to take into consideration to assign a modulator treatment to certain patients is allelic expression imbalance or allelic skewing in heterozygous. So we evaluated two patients, heterozygous patients, and on the left side, the patient heterozygous for G551D and F508L. We observed that the allele G551D is predominantly expressed. So these results show that the allelic imbalance can also have an impact in the right choice of the modulator treatment. When we think of the tests that we run to evaluate therapies in vitro, we need to consider that certain mutations can behave different in this respect. So we observed that mutations G551D and W1282x, the corrector molecules BX09, here, or BX661, may stimulate activation resembling potentiator activity. And also the level of CFTR activity, in the cell membrane can also be impacted by the modulator treatment. And the way it has been impacting certain other mutations is different than it was described for the most common mutation 508L. For instance, in the variants N1303K and W1282x, we observed that the potentiator BX770 can increase the level of CFTR protein localized in the cell membrane. And moreover, it was published that the variant F508L exhibit a lower level of CFTR protein after co-treatment with a BX770 and BX09 compared to the treatment of BX09 alone. That what we show here that other variants, in this case W1232x, the BX770 helps BX809 to increase the steady state level of CFTR protein. So these results are another indication that we need to evaluate more variables to enable more precise predictions of the modulator response. And for the summary of this presentation, so in primary airway epithelial cells, a benchmark of 30 to 40% wild-type CFTR chloride current has sometimes been used to predict clinical response to modulators. The magnitude of short-circuit current of the wild-type CFTR is variable and occurs across a significant range and should be considered when making predictions of this type. The mRNA levels for wild-type or rescue F5ODL CFTR expressed in primary airway epithelial cells can vary by about 300% and correlate with CFTR mediated CFTR transepithelial transport. A standardization of mRNA expression is likely to represent an important factor for predicting clinical response. Allelic expression imbalance has been associated with phenotypic variation, but has not been adequately evaluated for CFTR. CFTR modulator treatments are often directed towards specific mutation on one allele. Investigation of both CFTR loci for evidence of allelic skewing may provide useful information relevant to modulator treatment. Certain disease-causing variants can exhibit unanticipated CFTR activation profiles, like potentiation, following acute treatment with correctors such as BX661 or BX809. Potentiators, including BX770, may elicit increased level of steady-state, surface localized CFTR protein following chronic dosing in FRT cells. Mutation-specific responses to correctors and or potentiator treatment should be more prominently considered when assessing personalized therapies for rare CFTR defects. And I want to thank to Dr. Shaughnessy for leading this project and to all members of Social Lab and to all these institutions listed here for providing the primary results for our studies. Thank you.
Candela, that was a very interesting talk and obviously very pertinent to exactly what we're discussing today. We have some questions coming in. So first, do you know the mechanism behind allelic skewing? Is the transcription rate different between the two alleles, or is the mRNA stability impacted by certain mutations?
Well, in this case, the mutation I'm showing here, for instance, G541D delta F, for sure it has to be the mRNA expression. But in case of the other patient I'm showing there, where it's shown PTC mutation, certainly it must have been somehow degradation of mRNA.
Thank you. And then we have a kind of a comment from Scott Rendell. CFTR is expressed in specific cell types in the culture method, and quality has a big impact on the presence of different CFTR expressing cell types, such as secretory cells and ionocytes. I wonder what your thoughts are on that.
Sorry, say it again, please.
Oh, yes, that the culture method and quality has an impact on certain critical CFTR expressing cell types like secretory cells and ionocytes.
Yes, but in our lab, we try to keep exactly the same type of culture and the same conditions all the time. Even the passages of the cells are always consistent. We try to do that in order to have similar response, at least to keep the same conditions consistent.
Yeah, and I do wonder, I was thinking about variables such as media and other things that can influence CFTR quality. So I guess I have a question. Are there consensus statements or guidelines to standardize these protocols between labs to more easily let us compare?
Certainly it should be, but I don't think there is anything like that. But as I said, in our lab, we try to be very consistent and using always the same medium for growing cells, and try to span the cells in the same until the same passages in order to keep at least that's what we can control at that point, right? Mm-hmm.
Great. Well, thank you for your work and for presenting your data. I think it's such an important consideration for all of us.
Thank you very much.
You're welcome. We will move on now to our third speaker, Dr. Kevan Shaughnessy from National Jewish Health, and we'll be discussing elexacaftor as a CFTR potentiator.
Hi, my name is Kevan Shaughnessy. I'm an NIH postdoctoral fellow under the supervision and mentorship of Pamela Zeitlin, and working in collaboration with Preston Bratcher here at National Jewish Health in Denver, Colorado. The talk I'm going to give today is entitled Elexacaftor as a CFTR potentiator, Synergism with Ivacaftor, and Implications for New Combination Drug Therapies for Cystic Fibrosis. This work has been recently published, and you can see the citation for that here on the screen. I also present a lot more data in the poster, which is number 609. Us co-authors have no relationships we need to disclose. So, as you are aware, cystic fibrosis is a genetic disease caused by mutations to the gene encoding CFTR, which is a chloride channel. The CFTR mutations are categorized across several classes, which reflect distinct functional defects in CFTR. The data I'm going to present today is going to focus on class 2 and class 3 CFTR mutations, although the manuscript covers class 2, 3, and 4 CFTR mutations. So current treatment for cystic fibrosis includes treatment with CFTR modulators. These are small molecules aimed at rescuing specific functional defects, such as folding, trafficking, and channel gating. CFTR correctors rescue channel protein folding and membrane trafficking, and CFTR potentiators rescue channel gating and conductance. Okay, CFTR correctors work on a chronic timeline, so more like hours to days, whereas CFTR potentiators have immediate effects, so within minutes. The mechanism of action of ivacaftor, which is a long introduced CFTR potentiator, and tezacaftor, a CFTR corrector, are relatively well understood. The double combination of ivacaftor and tezacaftor is known under the brand name Symdeko. Now, more recently introduced has been Trikafta, which is a triple combination therapeutic of ivacaftor, tezacaftor, and elexacaftor. Elexacaftor being introduced as a CFTR corrector, an additional CFTR corrector in this treatment regimen. Now, given the substantial improvement in clinical efficacy of Trikafta over Symdeko, there is quite a lot of interest in more fully understanding the mechanism of action of elexacaftor. So that was the goal of this work, to further investigate the CFTR modulating mechanism or mechanisms of action of elexacaftor. In particular, we were interested in the potential role of elexacaftor as a CFTR potentiator. So in our lab, we use primary derived human nasal epithelial cells as model. And these cells are derived from donors with and without CF. And what we do is we expand and allow these cells to redifferentiate at an air-liquid interface, mimicking the same environment of an airway. And we allow this differentiation over about 21 to 28 days. And then we measure CFTR function in an Using chamber using an electrophysiological approach. Here's a typical experiment from our laboratory. In this case, these cells were derived from a donor homozygous with delta F508 CFTR. So we can apply drug treatment either chronically or acutely. Our chronic drug treatments are typically 24 hours. So in this experiment, we drug treated chronically with DMSO, which is just a control, Symdeco, which is the combination of tezacaftor and ivacaftor, and Trikafta, the combination of tezacaftor, ivacaftor, and elexacaftor. Panel A here shows a typical readout from the Using chamber. And what we're showing is short circuit current, which is indicative of ion transport. So the greater the short circuit current, the more ion transport occurring. So you can see at baseline here, before we apply any drugs acutely, the cells that were treated with Trikafta have a higher short circuit current. They are transporting more ions than the cells treated with Symdeco or DMSO. Forskolin and IVMX are in vitro activators of CFTR. They raise intracellular CAMP. And so when we apply Forskolin IVMX acutely, we get an increase in short circuit current because we are activating any functional CFTR. You can see that the increase in short circuit current after Forskolin IVMX application is greater in the cells treated with Trikafta than that of the cells treated with Symdeco or DMSO. And then CFTR inhibitor 172 is a specific inhibitor of CFTR. And so when we apply that acutely, we get a dramatic decrease in short circuit current. That's because we are shutting off ion transport by inhibiting CFTR. And again, you can see that the change in short circuit current is much greater in the cells treated chronically with Trikafta compared to Symdeco or DMSO. Panel B shows that quantitatively. What we're showing here is the change in short circuit current after treatment with either Forskolin IVMX or CFTR inhibitor 172. And the takeaway from this figure is just how powerful the incorporation of elexacaftor into that drug combination is. So the cells treated with Trikafta show much greater change in short circuit current in response to Forskolin IVMX and CFTR inhibitor 172. So that means that the CFTR function is much greater in the cells treated with Trikafta than those cells treated with Symdeco or DMSO. So a key observation we made at the beginning of this study that sort of led to further investigation was that elexacaftor, we know it's a CFTR corrector and works on a chronic timescale to rescue delta F508 CFTR function. But a key observation we made is that it behaves differently than other CFTR correctors on an acute timescale. So here, the figure presented here shows data in non-CF nasal epithelia. When we apply CFTR correctors acutely at increasing concentrations, we see no effect of lumacaftor or tezacaftor, which are both CFTR correctors. No effect relative to DMSO, but elexacaftor clearly increases short circuit current. It behaves differently than other CFTR correctors. So our hypothesis was that in addition to its known role as a CFTR corrector, elexacaftor is also a CFTR potentiator. So the rest of the data I'm going to present studies in human nasal epithelial cells derived from a donor homozygous for G551D CFTR. So this first experiment, there was no chronic treatment. Okay, so everything here was done acutely. So G551D CFTR is trafficked to the membrane, but dysfunctional on the membrane. It requires a CFTR modulator to function, a CFTR potentiator to rescue channel gating. And that potentiator has been ivacaftor. Ivacaftor is quite effective at rescuing function of G551D CFTR. So in our experiment here, we had no chronic treatment, but we acutely treated with ivacaftor or elexacaftor, or the combination of the two, and measured CFTR function. So in panel A here, you can see that full electrophysiological recording, that full experiment. The thin black line is G551D cells that were just hit with DMSO acutely, so no ivacaftor or elexacaftor. The gray line is a recording of cells that were hit with ivacaftor, but no elexacaftor. And then the purple lines show the combination of ivacaftor and elexacaftor. So the thick purple line shows a first application of ivacaftor and then an application of elexacaftor. And then the thin purple line shows the reverse. First an application of elexacaftor, then an application of ivacaftor. So what we can see in this experiment is that when you hit with the CFTR activators, forskolin and IBMX, you see no apparent increase in CFTR function, not much, because G551D is functionless unless it's rescued by a CFTR potentiator. Then when you apply ivacaftor, you get a nice increase in short circuit current because you are rescuing that channel function. Interestingly, you can see that the greatest increase of short circuit current was from those epithelia treated with the combination of ivacaftor and elexacaftor. In the two figures to the right, I want to more sort of intimately show how these two channel CFTR potentiators interact, okay? And that is synergistically. So in panel B, you can see the response to ivacaftor in the absence and presence of elexacaftor. So on the left in the gray bar is the response to ivacaftor in the absence of elexacaftor. Okay, and in the purple bar on the right, you can see that the response to ivacaftor is much greater in the presence of elexacaftor. And then in panel C, we're showing the response to elexacaftor in the absence and presence of ivacaftor. So you can see the bar on the left here, the purple bar is the response to elexacaftor in the absence of ivacaftor, and then the dash bar on the right is the response to elexacaftor in the presence of ivacaftor. It's a lot of caftors. So clearly what we're seeing here is that the response to either elexacaftor or ivacaftor is greater when introduced in the presence of the other potentiator. So these modulators are co-potentiating synergistically G551D CFTR. And when we use the CFTR inhibitor 172, we see that the greatest CFTR function was achieved when these two were applied together compared to ivacaftor alone, or of course DMSO. And that the order of application didn't matter. In this last experiment, we are looking, instead of acute application, we're looking at chronic application of ivacaftor and elexacaftor. So this is again in G551D CFTR epithelia. So the thin black line is again just DMSO, so cells treated just with DMSO. The thin purple line is also cells treated with DMSO, but we hit acutely with ivacaftor and then elexacaftor. And then the thick lines are cells treated chronically with drugs. So the purple line is the Alexa-Iva combination, and so just elexacaftor and ivacaftor double combination. And then the red line is Trikafta, the tezacaftor, elexacaftor, ivacaftor combination. And what we can see here is when we activate with forskolin IVMX, we get a dramatic increase in short circuit current in the cells that were chronically treated with drugs. And that we can achieve the same short circuit current if we apply elexacaftor and ivacaftor acutely. And then the graph here on the right is just showing that total CFTR function as measured by the CFTR inhibitor responsive short circuit current. And you can see that elexacaftor and ivacaftor achieve the same CFTR function that Trikafta does. Okay, so in conclusion, we've described a novel mechanism of action of ivacaftor, not as a CFTR corrector, but as a CFTR potentiator. And a couple other studies that I've noted here have also observed this. Interestingly, when used in combination, elexacaftor and ivacaftor co-potentiate synergistically mutant CFTR. And that this potent co-potentiation by elexacaftor and ivacaftor may be the mechanism of action underlying the efficacy of Trikafta It has recently been shown for gating and residual function CF genotypes. So thank you for listening. This work was funded by the National Institutes of Health and the Cystic Fibrosis Foundation and National Jewish Health. Yeah. So thank you.
Thank you for that description of some unexpected actions of elexacaftor. And although we are aware of continued choppiness of the video, we do apologize for that. I would encourage you to reach out over the conference website to any speakers if you have had unanswered questions you'd like to follow up on. So for now, to get us started for this discussion, Kieran, I was wondering, it doesn't seem like we have any indication that tezacaftor is harmful or beneficial in at least the G551D population. I wonder what your thoughts are on, since the only formulation available is a triple therapy with Alexa, Teza, and ivacaftor, what your thoughts are on if this is the best formulation.
Right. Can you hear me? Just want to make sure.
Yes, we can.
Okay. Yeah, good question. Right. The triple combinations obviously effective, includes ivacaftor and elexacaftor, and whether tezacaftor is harmful, I think is I'm sure minimally important. I doubt it, and I think what we're showing here is that the active compounds are really elexacaftor and ivacaftor. We know ivacaftor should be important because it's directly targeting the functional defect, but why? The question is why, I guess, was the incorporation of elexacaftor and tezacaftor helpful to rescuing G551D function, and thinking about tezacaftor and elexacaftor only as correctors, it wouldn't necessarily make logical sense. But now that we sort of are seeing elexacaftor more as also a potentiator, it sort of I think it explains that. And we were talking to some clinicians. I gave this talk at Children's Hospital here in Denver, and they said this is useful because it can help explain to parents why making the switch from an ivacaftor regimen to a trikaftor regimen would make sense for their children.
Yeah, it absolutely, I agree. It has important clinical implications for our patients who have gating mutations, but no delta F508 mutations, and what therapy would benefit them the most. Another question coming through, in the study of the G551D homozygous nasal epithelial cells, the tracings of VX770 and VX445 treated monolayers didn't exhibit complete inhibition by inhibitor 172. And wondering if you could speculate on what might explain this behavior.
Yeah, that is a good question and a very keen eye. We are not sure. I think this requires further investigation. If there's some secondary targets or other modes of ion transport being affected after the rescue of CFTR in some sort of co-regulatory way. These are all questions to answer, and if indeed it's increasing ion transport, chloride transport via some non-CFTR mechanisms because of some co-regulation by having a functional CFTR now on the surface. These are all important things to explore, but could be a reason that potentially other ion transporters become important once you rescue CFTR on the surface.
Right.
Could be a reason why we didn't get full inhibition, but more work needs to be done.
Thank you. Another questions, have you explored lumacaftor or tezacaftor to use in a higher dose range, such as a micromolar range, in the study of comparing acute potentiation abilities of luma, teza, and elexacaftor, as the EC50 concentration of these compounds can be far from each other?
Yes, that's a good point. Potentiators, I think, in a classical idea of them are that they have low EC50s there. They work on a quite sensitive scale to potentiate CFTR. So we, I think, both on the physiological relevance of it and just seeing where the effects and the saturating effects of elexacaftor were in those lower concentration ranges, didn't feel the need to look at much greater concentrations, because we're already seeing that divergence of function of elexacaftor from the other correctors. But certainly, if you apply much greater concentrations of other correctors like lumacaftor, tezacaftor, you maybe could see that. We haven't done that, so I don't know if you would. I think theoretically you shouldn't, because they're not potentiators. But if they're not working at that lower concentration range, I think that's all we need to know, is that elexacaftor is behaving differently.
We have a question on how are you defining synergistic versus additive activation?
Yep, I think simply just synergistic is greater than the sum of the two, greater than the sum of each individually, and synergism is synergism is greater than the sum of each individually. Additive would be the sum of each individual.
Well, thank you very much. Very pertinent talk, and one that certainly has important clinical applications. We'll move on now to our fourth speaker, Norman Allaire from the CFF laboratory in Lexington. We'll be discussing characterizing PTC mutation-induced aberrant CFTR mRNA splicing liability.
Hello, my name is Norman Allaire. I'm the head of genomics at the CFF laboratory in Lexington, Massachusetts. And today I'm going to talk about characterization and quantification of PTC mutation-induced aberrant CFTR mRNA splicing liability in both immortalized and in primary cells. Since we submitted the abstract, we've also had some additional information on correction of some of the splicing defects that we found and the functional consequence of those. So I'm going to include those towards the end of the presentation. I have no relationships disclosed related to this presentation. And so the CFF laboratory is focused, one of the focuses of the CFF laboratory is the discovery of read-through agents that will be beneficial for CF patients with PTC mutations. However, there have been reports in the literature of PTCs having secondary defects that relate to splicing and mRNA processing. And one of those was R553x a number of years ago that was reported to cause exon 12 skipping. So this is of obvious concern for read-through efforts because it can reduce read-through competent template, and that can inhibit or create issues for read-through therapeutics. So what we wanted to know was, we wanted to know if R553x induces any kind of splicing defect, as well as any of the other more common PTCs. So last year we presented some of this data. We looked at in silico analysis of these 5 PTCs, and we found that in that analysis that 4 of the 5 were predicted to impact splicing. And then we did isoform profiling in the isogenic line, the 16HB isogenic line and the 5 gene edited line shown here, and characterize some splicing defects. And so this year, what we want to do is we want to ask if those splicing defects, if they exist in patient populations, and we want to quantitate the fraction to understand the magnitude of those changes. But before we get into that data, what I want to do is just want to do a quick recap on what we've known about some of the major splicing defects that we found. And so the first thing I want to talk about is defects that are related or associated with R553x and G542x. And these are primarily exon 12 skipping events. Both of these mutations exist at exon 12. And so I'm showing you long-read, full-length long-read isoform profiles from 16HBs in the wild type and the 5 PTCs. And these are all treated plus and minus the SMIG inhibitor, which blocks editing. And so what you can appreciate is if you look at R553x, you see an increased fraction of orange, which is representing 12 through 15 skipping events, and an increased fraction of gray, which is 10 and 12 through 15. And then finally, we see presence of missing 12 or skipping 12. And if we look and compare, the next one is G542x, and we ask about defects in that, and there's obvious defects, again, relating to 12 to 15, inflation of 10 and 12 to 15. Those fractions are increased relative. So if we shift gears and we ask about splicing defects, R1162x and W1282x, in the previous graphs, we didn't show any defects related to these mutations, but that was with long-read full-length cDNA isoform method that uses primers to the first and last exon. Because they were predicted to impact splicing, we thought that maybe there'd be truncation events which wouldn't be picked up with those assays. And so there does exist a alternative polyadenylation and cleavage site in intron 22. And on either exon, there are either 1162x or 1282x. When this alternative polyadenylation signal is used, it results in a truncated CFTR, which truncates at exon 22 plus 9 amino acids, and then generates an alternative UTR. And what we've seen is that in both R1162x and W1282x, both of those, both the amount of the truncated message is inflated relative to the length. And that's what I'm showing on the right-hand side. So now this is long read, 3' race with long reads. And what we see with R1162x and W1282x is the percent truncation relative to full length is inflated in these genotypes relative to wild type or an upstream PTC G542x or a downstream missense mutation. Okay. So with that background, I want to talk about quantifying those fractions. And so we switched assays now. We moved to something droplet digital assay that gives us absolute quantitation. However, we need to correct for an RT bias that's demonstrated here. So what I'm showing is a plot of a series of droplet digital assays on the x-axis and the absolute copies on the y-axis. And if you look at the gray bars, this is absolute copies generated from a fixed amount of wild-type plasmid. However, if you take that same wild-type plasmid, transfect it into FRT cells, and then extract the RNA and then do RT-ddPCR, reverse transcription PCR, again using the same amount and source of RNA, what you see is varying copies of varying absolute copies of the transcripts, or the varying absolute quantities. Now, this is a problem because if we want to make comparisons from one exon, from one probe to the other, it obviously creates a bias. Luckily, because we have this onboard control, we can calculate or normalize out this variance or this difference, and we can correct for this. So that all the data, the droplet digital data I'm showing next is going to be mRNA droplet data that has been corrected for this bias. Okay. So now we're moving into primary cells. And so if we look on the left-hand side, the plot is percent exon skipping with this droplet digital assay corrected. And I'm showing you a donor, wild-type donor, and then a R553x G542x compound heterozygous from an intestinal organoid cell line. And what you can see is that in the donor plus or minus SNP, you have about 10% the skipping that just happens. However, when you look at the compound PET, with the SNP inhibitor, it jumps up to about 25%. And then with the vehicle treated, it goes up to about 35-40%. Now, if we switch over to the right-hand plot, rather, and we look at 3' percent 3' truncation as by this formula, again, with droplet digital corrected, what we see is with HBEs, we see that we have 2 independent donors plus or minus SNP. And what we see is a small fraction, about 10% with SNP, 5 to 10% on the vehicle for wild type. However, if we move into a W1282x homozygous HBE, what we see inflation up to about 35% with SNP and about 44% with vehicle. And then the story is very similar when we move into intestinal organoids. And while the magnitudes are a little bit lower, what we see is in donor, we see about 3 to 5% plus or minus SNP for wild type. However, when we move into 2 independent donors and IOs, what we see is about 12 to 15% in SNP treatment, and then that inflates out to about 40 to 42%. But I should say that the vehicle treated are being driven largely by NMD and reduction of that full length. So, now, what we want to know next is that we have identified this inflation of this truncation, and what we want to know is if we could disable it, does it provide any benefit? And so we rationalized that if we inhibit APA usage, we're going to reduce the amount of the truncation, and that should improve the amount of full length, and that could be used for read-through therapeutics. That could be beneficial for read-through therapeutics. And so what we did was we worked with our gene editing group, and what they did was they gene edited the R1162x 16HBE line, and they created a 83 base pair deletion at this primary APA site in the intron, so they didn't touch any of the exons. And with this, they generated a series of clones from this line. And then our question is, does that result in increase in full length? And does that, if it does that provide any benefit in functional assay? And so what I'm showing here next are the results of those gene editing experiments, and the results of the gene editing experiments. And I'm showing you the ratio of the truncated message compared to the exon 22, 23 junctions, truncated versus properly spliced. And if you look at the first is wild type, it's around 10%. When we go to R1162x, inflates to about 17%. And when we gene edited that 83 base pair deletion, what we see is a reduction back down to wild type levels. And I should say that this is driven by not only an increase in full length, but also a decrease in the truncation. So the next thing we want to know, is there any functional consequence for this? So we worked with our electrophysiology group, and what they did was they took these clones and they put them into a functional assay, and then we treated them with various read-through cocktails. And the read-through reagents we used were G418, which is aminoglycoside that promotes read-through, C9009, which is a degrader of ERF3A, which is a translational termination factor, and then the SNIG inhibitor again, which is a blocker of NACFC. And so what you can see is in the parental R1162x in yellow versus the deletion clone in purple, with every combination, whether it's G418 alone, G418 with SNIG, G418 with C9009, or all three together, we see a benefit, which is statistically significant. And so this suggests that there is really a benefit from this whole message that we're changing. So it's a proof of concept. We modulate the message, and then we get some additional function. So just to summarize, we've shown that the CFTR PTCs R553x and G542x have an additional processing liability of exon 12 skipping. We've shown this both in the 16HBE and primary lines. We've shown that W1282x causes an increase of an intronic APA site that results in alternative cleavage and polyventilation, and the upshot of that is the truncated mRNA, and we've shown this in primary airway as well as intestinal cells. And then we've shown that if you gene edit delete the primary APA site out of the 16HBE R1162x line, that not only reduces the amount of truncation, but it promotes the amount of full length. And furthermore, that increase in full length leads to an increase in CFTR function. So taken together, we believe that this data suggests that modulating CFTR mRNA processing may have therapeutic potential for the treatment of CF. And so I want to just conclude by thanking the team for all their work on this project. First, I'd like to start by thanking Martin Mentz, who leads the CFF lab. Thanks very much for his help and his support. My manager, Cal Cotton, as well as Ben, a great mentor and this project. Help to me, Herman Buehler, who runs the electrophysiology as well as the cell bank group for great scientific discussions. Hillary Valley, who runs the gene editing group, for all her work and her team's work for those deletion assay, creating those deletion clones. Emily Wilson from my team, who was recently gone on to graduate school, studying neuroscience, a PhD program in neuroscience. Andrea Pan, Jay Yoon, Matt Armstrong are other members of my team, and these guys were all responsible for the genomics work that's been done. Andrei Savichenko, who leads the bioinformatics group, and is my co-lead in bioinformatics and genomics. His team, Josh Conte and Barbara, in informatics would help with some analysis. Kevan, who leads the electrophysiology group, and Yi Cheng, who ran all the electrophysiology assays. And so with that, I think I'm actually a little bit ahead of time, but I will conclude, and I'm looking forward to discussions about that. Thank you so much.
Fantastic presentation. Thank you. I do want to note for everyone watching that with the ongoing issues, these will be fixed on the back end. So I do encourage you to rewatch this session as a playback to see these excellent presentations in their full. Norman, to start us out, as a person not in this field, I am just struck by the increasing complexity of addressing these nonsense mutations. And it appears from this work that there will be no stop mutation agnostic treatment, that you may in fact have to individualize treatments for particular genotypes. What are your thoughts on that?
So I think that's definitely possible. I think that I don't know if we fully appreciate the complete liabilities for all these PTCs. And I think they have different liabilities depending on where they live. So I think that's definitely possible. I think that for some of the PTCs, we've shown that the missplicing liability is larger than others, so it may be more relevant for some than others. But in general, I think that there's an awful lot we don't know about splicing and mRNA processing, and that's just an additional layer on a very complex system. So yeah, so I'm hopeful that we're going to be able to address some of it. But there'll be certainly will be cases where we'll need to have additional therapies specific to a PTC.
Would you anticipate any person-to-person variability in how much of these aberrant mRNAs are produced? It looked pretty consistent in the data that you showed today.
Yeah, so that's a really, really good question. And most of the data that we showed, we do realize that there are different patient-to-patient liabilities or different landscapes within each person's CFTR sequence. And that certainly is going to give rise to, certainly potentially could give rise to different splicing impacts. What we did for a lot of this work is we started off in 16 HBEs to have a uniform genetic background, specifically so we could see if we could identify these issues. But I think you make a very good point that because of these different liabilities, I think it has to be, we felt it was very important to validate them in a series of primary samples from different patients. So yeah, it's not a completely straightforward answer. I think that the human genome is incredibly complex, and patient-to-patient variability is a real thing.
Yeah, absolutely. It just reinforces the importance of this work as we move towards therapies for these nonsense mutations. Another question coming in: have you explored whether G418 could modulate exon skipping patterns in early or late PTC mutations?
That's it. We haven't. I know it's been looked at in the lab. I've joined about 4 years ago, so I haven't been specifically involved in that work. But that's something that I think the lab has looked at. I know that there are differences in read-through efficacies, or I've heard there were differences in read-through efficacy in early and late PTCs, but I haven't personally done that work.
Obviously a very complex and exciting issue. Thank you. And we'll move on now to our final speaker, Suma Traore from University of Iowa. We'll be discussing methods of enhancing delivery of CRISPR-associated nucleases and adenine base editors to airway epithelia.
Hi, everyone. I'm Suma from Dr. Paul McCrae's lab at the University of Iowa, and today I'll be talking to you about how we've enhanced delivery of CRISPR-associated nucleases and adenine base editors to airway epithelia through iteratively designing and screening amphibolic peptides. There are no relationships for me to disclose related to this presentation. So in the lungs for CF, aberrant transport of anions across epithelial surfaces leads to the trapping of mucus or mucus lungs that ultimately increase susceptibility to infections and inflammations, which result in progressive lung damage and lung disease over time. And so to correct the CFTR gene, we can modify these mutations with therapeutic proteins or genome editing reagents such as CRISPR or base editor RNPs. But one of our hurdles is effectively delivering these therapeutic reagents into airway epithelia. So in collaboration with Feldon Therapeutics, we've come up with a strategy for delivery using amphiphilic peptides. Our shuttle peptides involves two domains, a cell-penetrating domain, which predominantly consists of cationic residues in the C-terminus. And this feature was originally derived from the cell-penetrating peptides to help transport proteins and macromolecules such as GFP, Cas nucleases, or editors across the plasma membrane. The second domain of our shuttle peptide involves combining the endosomal linkage domain to the cell-penetrating domain via a linker as a means to prevent endosomal capture of molecules to further enhance our delivery. So here I'm showing you that the cell-penetrating domain of the shuttle peptide interacts with and disrupts the plasma membrane through hydrophobic interactions with the hydrophobic lipid tails of the membrane to help translocate molecules into the cytoplasm and then into the nucleus. And here I'm showing you that the shuttle peptide quickly disrupts the plasma membrane and delivers GFP protein into the cytoplasm within 45 seconds. And then within 2 minutes, the GFP protein is imported into the nucleus where it resides for the next 24 to 72 hours. And this demonstrates that the shuttle peptide provides both rapid delivery and rapid onset of effect. So the first generation of shuttle peptides that we delivered fused together the endosomal lytic peptide CM18 and the cell-penetrating peptide PTD4. And this CM18 PTD4 peptide effectively delivered various cargo into multiple cell types. And so the next steps involved modifying its peptide sequence to improve delivery. And so here I'm just showing you a subset of these modified peptide sequences, S10, S18, and S85. And this subset of shuttle peptides, when you compare it to the original CM18 PTD4 shuttle peptide, differ in cationic residues, hydrophobicity, and peptide length. And so to test their activities, we decided to transfect natural killer cells using Cas12a as our genome editing protein as a proxy for the hard to transduce airway epithelia. And as you can see, the novel peptides improved editing efficiency when you compare it to the CM18 PTD4 peptide. And so with this data, we hypothesized that these shuttle peptides could deliver proteins and Cas RMPs to airway epithelia. And so to test this hypothesis, we used human primary cells, airway epithelial cells grown at an air-liquid interface. We delivered Cas12a and Cas RMP with the various novel shuttle peptides in human airway epithelia. And we observed that the S10 shuttle peptide was the most efficient in Cas12a RMP delivery and achieved similar levels of editing with Cas9 RMP delivery. And we did so by measuring, figured this by measuring the percentage of insertions and deletions or indels through a PCR-based assay. And so in addition to identify the cell types that were transduced in the human airway epithelia, we also did some immunohistochemistry with GFP protein, and we see that the GFP enters and co-localizes with ciliated and non-ciliated cells. And after seeing our in vitro results, our next steps involved using the double fluorescent Cree reporter mice, ROSA 26, to measure editing efficiencies of the Cas RMPs delivered using the S10 shuttle peptide in vivo. And so we classified cells to be edited when we saw GFP positive cells as a result of the Cas RMPs targeting their respective LOXP sites and deleting the TD tomato cassette. And we were able to see delivery of each cargo into the large and small airways of our mouse model and quantify the editing efficiencies. And so overall, from the paper that we published in 2019, we were able to establish the conclusion that the shuttle peptide S10 delivered the Cas nucleases into the human airway epithelia and facilitated editing in the lungs of our transgenic mice. And with the S10 established as the peptide with the best editing efficiency in vitro and in vivo, the next question that we wanted to answer is, can we improve the S10 shuttle peptide to improve delivery and editing efficiency through an iterative screening process? And so to answer that question, we went back to the S10 shuttle peptide, and with our collaborators at Feldon Therapeutics, through an iterative screening process, we optimized the three different regions of the shuttle peptide: the endosomal linkage domain, the linker, as well as the cell-penetrating domain. The parameters for our optimizations included adjusting the charge of the N and C termini, altering the hydrophobicity of the N terminus, and switching the amino acid positions and so forth. And by modifying these various regions, we could potentially enhance our delivery of the Cas RNPs to airway epithelia. Once these modifications were completed, Belvin sent us the novel shuttle peptides in an aqueous solution that we then combined with CRISPR RNPs for apical delivery to the human airway epithelia at an air-liquid interface. After 3 days post-transfection, we harvested the genomic DNA and we used the samples as a PCR template in the first PCR or PCR1 to amplify the targeted locus of interest. And then we diluted the PCR products in PCR1 and then used that as a template for PCR2 to attach unique barcodes or index sequences for next generation sequencing via the MiSeq instrument. Once the sequencing was complete, we were able to get these FASTQ files that we then deconvoluted to analyze each sample. So here's the data with the Cas9 nuclease screening peptides. And on the x-axis, we have the number of experiments we conducted. And within each experiment, we screened 10 or more peptides on at least 3 different non-CF donors. And on the y-axis is the percentage of indels created from the non-homologous end-joining DNA repair mechanism, which occurs when the Cas9 RNP produces the double-stranded breaks in the DNA. This percentage of editing is how we quantify the delivery and editing efficiency. And so after each experiment, we examined the peptide sequences with high indel percentages or delivery efficiencies and optimized it through the various parameters that I mentioned before to create novel shuttle peptides to assay as we moved along. And as you can see with each iteration of peptides, there's an upward progression in the percentage of non-homologous end joining and delivery efficiency. In this next graph, on the x-axis, there are 2 subsets of peptides. On the left are peptides that we previously screened, and on the right are peptides that we newly designed through the iterative screening process. And I'm highlighting the shuttle peptide S10 because, again, in our previous work, that was a peptide that showed the most promise, not only in vitro, but also in vivo. And as you can see through our screening process, we were able to improve the delivery efficiency of the Cas9 RNP into our human airway epithelial cells by 35%. After seeing the in vitro results with our novel peptides, naturally, we wanted to look in vivo to see if the delivery efficiency and the editing efficiency increased as well. And so this experiment was conducted at the Small Animal Testing Center as part of our somatic gene editing consortium project. And here are some images comparing the S10 shuttle peptide with one of our best shuttle peptides, D237. And as a reminder, before I get into the quantification of editing, the mouse model that we used was a double fluorescent Cree reporter mouse for Rosa 26. And we classified cells to be edited when we saw GFP positive cells or green cells as a result of the Cas9 RNPs targeting their respective LOXP sites and excising out the TD tomato cassette, leaving behind that GFP cassette to be expressed. And for today, I'll only be showing you the 237 data because we don't have the S10 data quite on hand yet. But as you can see, we achieve editing in both the small and large airways in both male and female mice. And on average, we observe around 16 to 25% GFP positive or edited cells. So far, our data has shown that we have iteratively improved the shuttle peptide S10 and increased delivery efficiency. And so our next project involves changing our genome editing tool from the CRISPR-Cas9 RNP to an adenine-based editor RNP. And the advantage of using an ABE or adenine-based editor instead of a Cas9 RNP is that we can change one base pair to another without producing the double-stranded break that is typically associated with that CRISPR-Cas9 system. So this means that we could precisely edit our target genome locus of interest without randomly inserting and deleting base pairs through the non-homologous end-joining DNA repair mechanism. In addition, base editors are independent of the cell cycle, unlike the CRISPR-Cas9 system, which suggests that we can edit cells that are meiotically quiescent like those found in the airway epithelia. Further, in addition to the advantages that I just mentioned, adenine-based editors as a new gene editing tool have the potential to correct about 46% of single point mutations that occur within the CFTR gene that are CF causing. So overall, ABEs serve as a promising instrument for future therapies. So to begin, I just want to give a brief overview of what adenine, about the adenine-based editor. And so you have and how it works. So you have an adenine nucleotide that is the desired target within the DNA region of interest. And then you have your complex that is composed of a guide RNA and a catalytically impaired Cas9 nickase that is linked to a deoxyadenosine deaminase. The Cas9 nickase linked with this deoxyadenosine deaminase is what we recognize as an adenine base editor. And so once you have this complex, the guide RNA is responsible for targeting and binding the DNA locus of interest. And once it binds, it displaces a small bubble of single-stranded DNA. And it's within this bubble where the deoxyadenosine deaminase converts the adenine to an inosine. Following that conversion, the Cas9 nickase recognizes the protospacer adjacent motif, or the PAM sequence, and nicks the strand opposing the edited strand. And so after this nick, the DNA uses the cellular repair mechanism, and it's within this repair that the inosine is recognized as a guanine, which ultimately replaces that original A to T base pair with the G to C base pair. So in some of our previous work, we were able to demonstrate the use of ABE RNPs as a tool to repair CFTR nonsense mutations. And so here I'll be finishing this talk by mentioning how we investigated the utility of amphiphilic peptides for delivery of ABE RNPs in human and non-human airway epithelia. So in the cells, we targeted the beta-2 microglobulin gene. In the human cells, we target the beta-2 microglobulin gene, and B2M is a small protein that is associated with the major histocompatibility complex, meaning that it's on the surface of all nucleated cells, and it's a great target. And so for the human non-human primate cells, we target the CCR5 gene because it's generally used as a safe harbor site for cellular disruptions, and it's a convenient spot for us to access as well. So here's the data quantifying base editing in human airway epithelial cells targeting the B2M gene. And so on the x-axis, we have the peptides we tested. On the y-axis, we have the percentage of base editing. The same is true when we target the CCR5 gene in rhesus airway epithelial cells. And we see, and as you can see, that we are able to achieve a base editing at different efficiencies using the amphiphilic peptides, the novel amphiphilic peptides, as a delivery strategy. And so our ongoing experiments involve delivering ABEs with peptides targeting the CFTR gene. So to conclude, we were able to yield peptides with higher editing efficiencies in the S10 in the human airway epithelial cells using our iterative screening process in vitro. And we were also able to demonstrate editing in airway epithelia in vivo using the peptides. Lastly, we're able to show delivery of ABE RNP with the peptide using the human and non-human primate airway epithelial cells. And for future directions, we would like to continue to evaluate the use of amphiphilic peptides to delivery of the base editor for future therapeutic applications. And we'd also anticipate assessing delivery of gene editing tools to the airways of animal models, such as the rhesus monkey and the pig, to critically evaluate the efficacy of these tools. And finally, we'd like to investigate the feasibility of peptide-mediated delivery of ABE to correct CFTR mutations, especially mutations that are currently non-responsive to any of the available cystic fibrosis treatments. And so for acknowledgements, I would like to thank my PI, Dr. Paul McCray, for his guidance and assistance, and for everyone in the Paul McCray Lab for their contributions and support. And I would also like to thank David Gee and Stephanie Halley on the field and therapeutics team and everyone associated with that team as well. Special thanks to David Liu and Greg Newby, as well as Mark Belkey and everyone at IDT. Thank you everyone for listening, and I will take any questions that you have.
Thank you, Suma. I have a question to start us off. Have you ever investigated the efficacy of these peptides in inflamed airway models or in animal models with lung disease?
No, currently we have not, but that is something on the agenda that we're looking forward to doing. We have some CF models that we're looking to, mice models that we're looking to deliver base editors to with our peptides.
And would you hypothesize that would provide additional barriers, or would the inflammatory state of the lungs be beneficial in any way?
I'm sure it will come with its own set of barriers. Of course, there's mucus that we would have to deal with. There's a number of other characteristics associated with inflamed lungs that we would also have to address as well.
Sure, and I feel like that's probably a universal issue for these types of delivery mechanisms. Another question came in. New data suggests that the best possible target for CFTR gene therapy is correcting the basal cells. Such correction would possibly establish a lifelong cure by targeting cells with the capacity to replenish the surface epithelia. Do you have any data showing that your peptide delivery method can reach and correct basal cells in a native in vivo environment, or other data?
We currently don't have any data to share regarding our peptides targeting basal cells. We do have some ongoing studies happening right now where we've delivered naphthalene to some mice models, just slewing away all of the epithelial cells of the surface, and see that we don't have any results yet for that, but we're investigating whether or not we are getting to the basal cells to see if they can get so that they can generate other cell types of the lungs. But it is an interesting question to see if we can get to the basal cells. Hopefully we can. Yeah. But there are, I mean, of course, in the CF, in the lungs, there are different types of progenitor cells across the lung tree. So seeing if we can get access to those progenitor cells also will be of interest.
Yeah, absolutely. I believe with your animal model, this was an intranasal installation. I wonder how you would see other methods of delivery and patient delivery in the future.
Yeah, I'm not sure how that would happen, maybe intraperitoneally, but I'm not too sure about that.
Another question, do you have data on whether the delivery peptide might induce any immune responses in animals?
Yeah, so in our previous work, we've looked at toxicity and looking at addressing cytokines and neutrophils and macrophages, and we don't see any differences for a mouse treated with PBS or a mouse treated with given peptides to deliver Cas RNPs. So we still have to address other immune responses, but currently that's all we have.
So optimistic that hopefully it would not provoke anything untoward with immune responses.
Right.
Well, thank you, Sumba, and thank you to all of our speakers today for their excellent presentations. Thank you to the audience for your patience. I'd just like to reiterate, if you have questions that were unanswered in today's sessions, please feel free to reach out to our speakers over the conference website. And also to remind you that in the playback video of this, they will try to fix the streaming issues and the technical issues that we've experienced today. So please do rewatch this so you can appreciate our speakers, and take care. And I hope the rest of your conference goes well.